In this lesson, we're going to talk about the first step of cellular respiration, which is called glycolysis, and also we're going to talk about a alternative to aerobic cellular respiration, which we've mentioned, which is the anaerobic process of cellular respiration. So whenever I'm near you, I undergo anaerobic respiration because you take my breath away. Really quick, we'll go through these 3 quick questions, so pause the video here. So the first question says, propose a hypothesis as to whether the bacterial precursor to mitochondria or chloroplasts form an endosymbiotic relationship with eukaryotic cells first. Okay, lots of tough words there, but precursor just means what came before mitochondria or chloroplast, and the endosymbiotic relationship is the mutually beneficial relationship that these had. Hopefully you can identify that most likely the bacteria that came as a precursor to mitochondria came first. And the reason why we know this is simply because plant cells have mitochondria and chloroplasts, and animal cells have only mitochondria. So it's more likely that mitochondria were taken up and then later on chloroplasts were taken up than mitochondria having been taken up twice. Okay. Question 2 says, prior to leaving the nucleus, mRNA gets modified. So we can see here in a process called splicing. What is the name of the sections of mRNA which are removed, and what is the name of the sections which remain in the mature mRNA? All right, the sections which are removed, you should know, are called the the introns, and the ones that remain are the exons. All right, and this is because introns stay in the nucleus, exons exit the nucleus. Okay. What is the name and function of the 2 nucleotide sequences added to the 5' and the 3' ends of the mRNA molecule? Hopefully you know that this is the methyl cap, and then this is a poly A tail. Okay, and their function is to protect the mature mRNA. Okay? Alright, so looking at cellular respiration, we want to know about where glycolysis happens and what are the inputs and outputs of glycolysis, including the ATP yield. Okay? We don't want to know the details of the biochemical pathway mechanisms, okay? And then we also want to know the location of anaerobic cellular respiration, its inputs, and the difference in outputs between animals and yeasts, including ATP yield, okay? So, as we spoke about last time, every living thing breaks down glucose. Okay, the little tiny glucose carbohydrate is the source of energy for every living thing in the world. So every living thing must actually break it down to get ATP. Okay, so glucose is really, really important, pretty much. The way in which living things actually break down glucose, it's what's going to vary, and that's what we're going to look at today. So the first process of cellular respiration, the first process of breakdown of glucose is called glycolysis. And if we break down the word glycolysis, glyco means carbohydrate or sugar, okay? And lysis means breakdown, okay? So glycolysis literally means the breakdown of a sugar, more specifically the breakdown of glucose. So it's the process of breaking down glucose in the cytoplasm, okay, this is where it happens, into 2 molecules of pyruvate. And whilst glucose is a 6-carbon molecule, pyruvate is a 3-carbon molecule, so that's why we get 2 of those, okay? And here is pyruvate, all right? That's one carbon here, there's a carbon here, and there's a carbon there, okay? So a small amount of energy is actually released when we do glycolysis. We get about 2 ATP, and then we also get 2 hydrogens being released, and these result in the formation of those 2 loaded NADH molecules. So here we see the process of glycolysis. Okay, we have that 6-carbon glucose molecule, right? And that's broken down into 2 pyruvates or pyruvic acids, right? And in that process, we have ADP converting into 2 ATP, and we have 2 NAD pluses converting into 2 NADHs. Before we keep going, I want you to notice that when we're talking about anything to do with cellular respiration, NADH will be the coenzyme used, okay? If we're talking about anything to do with photosynthesis, NADPH will be the coenzyme used. Best way to remember that, photosynthesis starts with a P, NADPH, okay? So what actually happens next depends on whether we have oxygen or not, right? Ideally for eukaryotic organisms with a mitochondria, obviously, oxygen is going to be present, so we're going to get aerobic respiration happening, right? And the reason for this is because aerobic respiration, remember, we're going to get a lot more ATP out of aerobic respiration. But if we don't have any oxygen present, or if the organism is prokaryotic, the 2 ATP produced in glycolysis is all the energy that we can actually take out from the glucose molecule. So we're going to keep going, and we're going to have anaerobic cellular respiration proceeding. And what another name for anaerobic cellular respiration is fermentation, right? And those 2 NADH molecules, pretty much, they have to return to their unloaded form so that further anaerobic cellular respiration can actually happen. Okay, so they have to go back to being NAD pluses. And we do this by converting those 2 pyruvate molecules into in one of 2 processes. Either we convert it into lactic acid, and this is what animals undergo. And then yeasts and plants actually undergo what we call alcoholic fermentation. So this is lactic acid fermentation. It's the simpler of the 2 processes. That initial glucose molecule undergoes glycolysis first, and we're aware of that. We have the ATP produced, and we have some NADH produced. And then the product is those 2 pyruvates, okay? And the 2 pyruvate is then converted into the lactate, right? And in this process, we're going to reverse the coenzymes NADH back to NAD, okay? Alcoholic fermentation only adds simply one little step in here. So the glucose, first of all, gets turned into the pyruvate in glycolysis, and we have that activating of those coenzymes. Okay, they become charged. But the pyruvate, before it actually becomes the final product, it first becomes acetaldehyde, right? And in that process, it loses 2 carbon dioxide, and then that acetaldehyde is then going to go on and become ethanol, and the NADH is going to convert back to NAD plus. Okay. So it's important that you have an understanding of the chemical and the word equations for these 2 processes. So lactic acid fermentation, if we go back here, what we have is glucose through glycolysis becoming lactic acid, okay? So all we're going to have is the glucose, all right? If there's no oxygen, okay, in the cytoplasm, okay, what it's going to do is become lactic acid. Alright, and then remember that we are producing 2 ATP. Okay, if we were to do the chemical equation for this, glucose is C6H12O6. Right? Lactic acid is CH3CHCOOH, and we also are getting to ATP there, right? Okay. Alcoholic fermentation, on the other hand, we have glucose fermenting, still in the presence of no oxygen in the cytoplasm. And this time, though, we are going to get ethanol, which is an alcohol, and we're going to get carbon dioxide, and obviously again 2 ATP, right? This time we have C6H12O6, that's glucose, becoming C2H5OH, okay, CO2, and 2 ATP. Okay. Just do notice though that we actually don't have balanced equations here, and so we will need 2 lactic acids here. We're going to have 2 ethanols here, and we're going to have 2 carbon dioxides over here. Alright,